Usually when we think about our internal organs, especially the ones tucked deep, deep away inside our bodies, we tend to imagine this, like, pristine, heavily guarded vault.
Oh, absolutely. Like it's completely sealed off.
Right, like the ultimate VIP room in a nightclub or something. You just naturally assume an organ like the pancreas is completely closed off, totally sterile, and, utterly protected from the dirty, microbe-filled outside world.
Yeah, and you definitely aren't alone in thinking that. I mean, that assumption has actually been the bedrock of traditional medical consensus for a really long time.
Really? Like decades?
Oh, for decades. The pancreas was considered an entirely sterile zone. The idea that bugs could be just, living in there was almost unthinkable to doctors.
But today, we are diving into a really dense, cutting-edge medical review that completely shatters that biological myth.
It really does. It flips the whole concept on its head.
So our mission for this deep dive is to uncover exactly how microscopic entities, and we're talking everyday bacteria, fungi, and viruses, are literally breaking into the pancreas.
And doing some major damage once they get there.
Exactly. We aren't stopping at the break-in. We're going to trace the exact step-by-step algorithm of how they hijack perfectly healthy tissue and rewrite its programming to create one of the deadliest forms of cancer on the planet, which is pancreatic ductal adenocarcinoma, or PDA.
Yeah. What's truly fascinating here is how the technology of high-throughput next-generation sequencing has just upended everything we thought we knew.
Right, because before we were just trying to grow them in petri dishes.
Right, precisely. In the past, if we couldn't grow a bacteria in a lab dish, we just assumed it wasn't there.
Mm-hmm.
But now, by looking at the raw DNA and RNA profiles of the tissues themselves, we've discovered that a healthy pancreas actually harbors a rich microbial population.
Wow.
Yeah, in fact, it's incredibly similar to the tissue of the duodenum, which is the very first part of your small intestine. So the VIP room, as it turns out, was never actually empty.
I just love that visual. So let's really get into the weeds here. I want to trace exactly how these microbes break in, how they set off this crazy chain reaction in our immune system, and finally look at the mind-blowing therapies doctors are using to fight back. Because, these aren't just gut bugs, right? They're migrating from all over.
Exactly. If we look at the bigger picture, the pancreas does have a low microbial biomass compared to a place like your colon. There are fewer bugs overall.
Okay, that makes sense.
But when someone develops pancreatic cancer, we see a massive catastrophic shift in who is living in that VIP room. For instance, there was this seminal study that found a 3,000-fold increase in fungi, specifically a genus called Malassezia in PDI tissues compared to a healthy pancreas.
Wait. 3,000 times more fungi?
3,000 times. It's a staggering explosion in population.
That is wild. And Malassezia is a fungus we normally associate with the skin, right? Like, isn't that dandruff?
That's the exact one, yeah. Malassezia globosa. It's a very common skin fungus, and it is finding its way deep into the abdomen. And we see the same thing with the mouth, too.
Like dental plaque.
Yep. Bacteria like Porphyromonas gingivalis, which, originates in the oral cavity and is the main culprit behind gum disease. It just shows up in these pancreatic tumors. We even see viruses like HPV making the journey.
Okay, so the obvious question for anyone listening is, how on earth are bugs from your mouth and your scalp getting into a deep abdominal organ?
It's a long trip.
Right. So when I was reading the source material, I needed a way to visualize this. So imagine a closed medieval fortress that's your pancreas. The researchers essentially mapped out 4 distinct secret tunnels that these microscopic invaders use.
I think that's a perfect way to visualize it.
Yeah.
So what's the first tunnel in your fortress?
Okay, let's call it the front door. This is the main pancreatic duct. The bugs literally travel backward from the gut, sneaking up through a biological gate called the sphincter of Oddi.
Right, going against the flow.
Exactly. Then you have the side door, that's the common bile duct, where microbes sneak in through shared plumbing connected to the liver.
Tsuki.
Very. Third, we have the highway. This is the intravenous route. Microbes from your gums or your skin get into your blood and just hitch a ride straight through your portal veins into the pancreas.
And the fourth one?
The fourth one is my favorite, the ghost walk.
The ghost walk. That is a brilliant way to describe transmural migration.
Right, because it's literally like a ghost walking through a wall.
It really is. This is a phenomenon where microbes phase right through the physical walls of the colon directly into the pancreas, simply because the two organs are, physically resting up against each other in the abdomen.
But wait, I have to push back on that first tunnel at the front door.
Sure, go ahead.
If we look at the sphincter of Oddi, it's a muscular valve, right? Its entire evolutionary purpose is to act as a one-way gatekeeper to prevent backward flow from the dirty gut into the clean pancreas. So how do these gut bacteria get past a locked muscular door in the first place?
It's a really great question, and it all comes down to initial inflammation.
Like damage to the door itself?
Sort of. Experimental models, like when scientists track these fluorescently glowing bacteria in live mice, show that the bugs don't just march into a perfectly healthy, closed-off pancreas easily. What happens first is early, perhaps even minor, pancreatic inflammation.
So the environment has to be primed.
Precisely. We call it a permissive environment. When the pancreas is inflamed, the sphincter of Oddi essentially malfunctions. The muscle becomes lax.
Oh, it loses its tone.
Exactly, it loses tone, which causes the intestinal fluid to reflux backward. So the inflammation itself is essentially holding the door wide open, allowing a massive surge of gut microbes to rush in and set up camp.
Okay, so just to make sure I'm wrapping my head around this initial stage, and for you listening at home, let's do a quick recap.
Sounds good.
The pancreas is absolutely not a sterile fortress. Microbes from our skin, our plaque-filled mouths, and our gut are constantly using these four routes, our ducts, our blood, and even phasing through tissue walls to invade.
Right, they're extremely resourceful.
Especially when a little bit of inflammation leaves the door propped open, it really makes you stop and think. I mean, have you ever considered that a common skin fungus, or the bacteria sitting in your dental plaque right now, could literally travel through your bloodstream to take up permanent residence in one of your deep internal organs?
It fundamentally changes how we view the interconnectedness of our body's microbiome. I mean, nothing is truly isolated, but once those bugs set up residence, we have to look at the clinical damage they cause. Right. Because they don't just sit there quietly enjoying the VIP room.
Right.
This is where we move from mere colonization to the actual step-by-step progression of cancer.
Because this isn't an overnight switch, right? You don't just get a fungus in your pancreas on Tuesday and then have full-blown cancer on Wednesday. There is a very strict grading scale here.
Exactly. It's a highly structured biological algorithm of disease. And it usually starts with pancreatitis.
Just general inflammation.
Right. Now, pancreatitis can be acute, meaning a sudden painful bout of inflammation. And interestingly, this acute phase is often triggered by infections, including fungi like Candida.
Wow, so fungi are driving the early stages too.
They can be, yeah. In fact, severe acute pancreatitis is sometimes managed by doctors using antifungals. But if the problem persists, it turns into chronic pancreatitis, which is a long-term smoldering inflammation that leads to permanent fibrosis or scarring of the tissue.
And as this tissue is scarring and struggling, the doctors start looking for physical warning signs on the scans, right? Like IPMNs.
Yes, intraductal papillary mucinous neoplasm.
That is a mouthful.
It really is. But basically, these are benign mucus-producing cysts that form inside the pancreatic ducts. They aren't cancer yet, but finding bacterial colonies inside these cysts is considered a major red flag by oncologists.
A warning sign that things are heading down the wrong path.
Exactly. From there, the tissue can enter a strict precancerous algorithm known as PANINE, or pancreatic intraepithelial neoplasia.
I really want to slow down here because the way PANINE is strictly graded is fascinating to me. It reminds me of a building inspector checking for code violations in a high-rise.
Okay, I like that. Let's walk through the grades using that.
Great. So the first stage is PANIN 1, right?
Yes. PANIN 1 is low-grade mild dysplasia. Dysplasia just means the cells are starting to forget their specialized jobs and they lose their uniform shape.
They're acting a little weird.
Yeah. At PANIN 1, the architecture is just starting to look a little abnormal under a microscope. It's subtle.
So PANIN 1 is like the building inspector finding a cracked window. It's a violation, sure, but the building is still entirely structurally sound.
But then you progress to PANIN 2, which is intermediate grade. Now you have moderate dysplasia.
More cracks?
Lots more cracks. The structural abnormalities in the cells are highly noticeable now. The cells are piling up on each other. And finally, if left unchecked, you reach PANIN 3.
The danger zone.
Exactly. This is high-grade severe dysplasia. It is the final, most advanced precursor stage before the cells officially become invasive cancer.
By PANIN 3, the load-bearing walls are warping and the foundation of the building is actively crumbling, which leads to the final stage, PDAC. The building completely collapses.
And PDAC pancreatic ductal adenocarcinoma is uniquely aggressive and difficult to treat.
Why is it so hard to treat?
Well, histologically, once the cells become PDAC, they surround themselves with something called a desmoplastic stroma.
Desmoplastic stroma?
Yes. This is a thick, impenetrable, dense, scar-like tissue that encases the malignant cells. It essentially forms a biological bunker, making it incredibly difficult for chemotherapy drugs or your own immune cells to get inside and kill the tumor.
It's just wild to conceptualize. So another quick recap here. You go from what feels like a minor stomach ache or general inflammation to these little cysts, all the way down this strictly graded Pennian decline from a cracked window to a crumbling foundation until the cells lock themselves inside a scar tissue bunker.
It's a very rigid progression.
It completely reorients how you view minor inflammation in the body. I mean, it's not just a fleeting symptom. It could be the very first domino falling in a highly structured algorithm of disease.
Which highlights exactly why early intervention during those minor inflammatory stages is so critical. Because once we hit that final bunker stage of PDA, the microbes we talked about, they aren't just innocent bystanders anymore. They are actively forcing the tumor to grow.
Okay, so this brings us to the deepest and, frankly, most terrifying part of our dive today, the mechanics of sabotage.
This is where it gets really crazy.
How exactly do microscopic bugs command human cells to build a tumor? What is the actual mechanism there?
Well, it all comes down to hijacking cellular signaling pathways. Let's look at direct growth first.
Okay.
Remember that skin fungus, Malassezia globosa, the one we found 3,000 times more of in the cancer tissue?
The dandruff fungus, yeah.
Right. So it activates a highly specific signaling pathway in the human body called a C3a MBL axis. MBL stands for mannose binding lectin. Normally, MBL is a protein that helps our immune system recognize sugars on the outside of pathogens. It's a defense mechanism.
It's what's it called this.
Exactly. But here, the fungus uses it like a hacked keypad. When the fungus activates this axis, it essentially stomps on the gas pedal, directly forcing the tumor cells to multiply and grow.
Oh, man. So the fungus can directly act as an accelerator.
Yes, but they also run this incredibly complex immune hijacking operation too, using a totally different fungus called Alternaria alternata.
Now, when I was reading the literature on this specific hijacking, the only way I could make sense of the cellular biology was to frame the entire mechanism like an intricate multi-stage bank heist.
I actually love that analogy. Let's walk through the steps of this heist.
Okay, so step one begins with that fungus, Alternaria alternata. What does it do?
It interacts with the pancreatic cancer cells and forces them to release a specific molecule called IL-33. In biology, we call IL-33 an alarmin. It is literally a chemical distress signal.
So, step one of the heist. The microbes are the masterminds who intentionally pull the fire alarm in the building.
Exactly. Now, that IL-33 fire alarm floods the area. It specifically wakes up nearby first responder immune cells. In the literature, these are known as ILC2 and Th2 cells.
ILC2 and Th2. Okay, I'm just going to think of them as the rookie security guards.
That works perfectly. So these rookies wake up in an absolute panic because of the fire alarm, and they react by releasing a massive flood of their own chemical messengers, which are called cytokines.
So step two of the heist. The fire alarm tricks the rookie security guards into panicking and handing over the master radio codes, those cytokines, to the wrong people.
And those radio codes act as a severe corrupting signal. They specifically target a group of neutral immune cells called macrophages.
Macrophages. Those are the guys that eat up dead cells.
Right. Normally, they are the garbage collectors of the body. In their neutral state, we call them M0 macrophages. But those corrupting cytokines force them to change their state, polarizing them into what we call M2 macrophages.
And what do M2 macrophages do?
M2 macrophages are designed to suppress inflammation and promote tissue growth, which is the exact opposite of what you want when there's a tumor actively growing.
Wow. So step three, the radio codes completely corrupt the senior security guards. They flip them from M0 neutral to M2 dirty.
Yep. And here is the final fatal blow. Those corrupted M2 macrophages start secreting a protein called TGF-beta.
TGF-beta?
Yes. This protein wakes up a whole new squad of specialized suppressor cells called MDSCs. And together, this corrupted team works specifically to hunt down and completely suppress CD8 plus T cells.
Oh, no.
And CD8 plus T cells are the heavily armed SWAT team of the immune system. They are the primary cells responsible for recognizing and killing cancer.
That is insane. So step four of the heist, the corrupted guards and their accomplices literally lock the real police, the CD8 T cells, outside the building, creating what researchers call an immunosuppressive tumor microenvironment.
It is a brilliantly sinister biological strategy. The microbes literally rewrite the immune system source code so that your own body builds a protective barrier around the tumor instead of attacking it.
Okay, I have to pause and push back on this because the human body is not stupid.
No, it's not.
Right. So if the CD8 T-cell SWAT team is locked out, why doesn't the immune system just recognize the problem and send like way more CD8 T-cells to break down the door? How does a single fungal infection manage to overpower our entire adaptive immune system?
That right there is the million-dollar question in oncology today. And the answer goes back to that desmoplastic stroma bunker we talked about earlier.
The scar tissue.
Exactly. The sheer physical density of that scar-like armor acts as a literal brick wall against the T cells. But it's not just a physical barrier, it's a chemical one too.
A chemical barrier.
Yeah, think about it. The microbes inside the tumor are constantly metabolizing and digesting things, right?
Eating and producing waste.
Exactly. They flood the surrounding area with their own waste products, microbial metabolites like branched-chain amino acids and short-chain fatty acids.
DCAAs and SCFAs.
Right. And when you combine those waste products with the TGF-beta protein from the corrupted macrophages, it creates a toxic chemical smokescreen. So the CD8 T cells physically cannot penetrate the bunker wall, and chemically they are paralyzed by the smokescreen before they can even mount an attack.
Okay, recap number 3. The heist is real. The microbes use the C3a MBL pathway to step on the gas pedal. Then they pull the IL-33 fire alarm, corrupt the M0 macrophages into M2s, and pump out a toxic smokescreen of metabolites to lock out the T-cell SWAT team.
That's exactly right.
That is utterly terrifying, but medically it is so deeply fascinating. So if you're listening to this and putting yourself in the shoes of an oncologist, if you know the mechanics of this heist, where do you cut the wire? Do you try to block the initial fire alarm? Or do you try to rescue the T-cell SWAT team?
And that line of thinking is exactly what is driving the massive counterattack in modern therapeutics right now. Because once you decode the algorithm, you can finally disrupt it. We are now targeting the microbiome itself to save patients.
Let's talk about that counterattack. When we look at the clinical evidence, what are the doctors actually doing and what are the outcomes?
Well, one of the most illuminating data points we have comes from looking at patient survival data. Researchers compared the microbiomes of short-term survivors of pancreatic cancer against long-term survivors.
And what did they find?
What they found was astonishing. The long-term survivors actually had greater microbiome diversity inside their tumors. And along with that diversity, a much higher infiltration of those cancer-killing CD8 T cells.
Wait, really? So having more types of bugs in the tumor is actually a good thing?
It sounds completely counterintuitive, but yes. In ecology, it's called competitive exclusion.
Competitive exclusion.
Yeah. A highly diverse microbiome creates a balanced ecosystem that seems to keep the really bad pathogenic cancer-driving bugs, like our skin fungus, in check. It maintains a healthy immune balance. And we've literally proven this in the lab using FMT.
FMT, that's fecal microbiota transplantation, right? Taking stool from one subject and putting it into another. How did that work here?
Well, in preclinical models, researchers took fecal samples from a human long-term survivor of pancreatic cancer and transplanted that microbiome into mice with pancreatic tumors.
And what happened?
The mice's tumors actually shrank.
No way.
Yes. The diverse human microbiome successfully recruited the T-cell SWAT team back into the mouse's tumor. Conversely, when they transplanted feces from short-term survivors, the tumors grew and those immunosuppressive cells multiplied.
That is just incredible. But what about directly attacking the masterminds of the heist? Like, can't we just wipe out that Malassezia fungus?
We can do that using highly specific antifungals. For example, using a drug called amphotericin B.
Amphotericin B, okay.
Right. When researchers use this to wipe out the fungal populations, it completely breaks that IL-33 fire alarm cascade we just mapped out. The corrupted macrophages vanish.
The smokescreen lifts?
Exactly. When you remove the fungi, the toxic smokescreen literally lifts. And suddenly, standard chemotherapy drugs like gemcitabine, which previously just bounced right off the tumor, they become significantly more effective because the immune system can actually step in and help fight.
Oh, that is brilliant. And it's not just about killing the bad guys, right? The research is also looking at introducing helpful compounds, too. I was amazed reading about microbial metabolites like TMAO.
Yes, TMAO is a chemical produced by certain gut bacteria. When researchers took TMAO and combined it with standard immune checkpoint inhibitors, which are modern cancer drugs designed to take the brakes off the immune system, it massively boosted the immune response, significantly reducing the tumor burden in mice.
Wow.
But perhaps the most futuristic approach we are seeing is the rise of living drugs.
Living drugs. Okay, you have to tell me about this because this sounds straight out of a science fiction novel.
It really does. Scientists are currently engineering living probiotics to do our dirty work. For example, they've taken a strain of bacteria called E. coli Nissl 1917, and they genetically engineered it to produce a pore-forming toxin.
A pore-forming toxin?
Yeah, a toxin that literally punches holes in the cancer cells. Or they take a lactobacillus bacteria and coat it in a functionalized gallium polyphenol network.
So they're weaponizing good bacteria.
Essentially, yes. These bacteria are literally programmed to act as Trojan horses. You introduce them to the body, they intentionally seek out and colonize the tumor, alter the chemical microenvironment from the inside, and provoke a massive CD8 T-cell attack from within the bunker.
Okay, this is all incredibly promising, but it raises a huge, kind of obvious question for me.
Okay, what is it?
If we know for a fact that these bad bugs are causing the cancer to grow, and we know that killing them helps the chemotherapy work, why wouldn't a doctor just prescribe massive, broad-spectrum antibiotics to every single patient at the very first sign of a pan-in-one cracked window? Just nuke the whole site from orbit.
Oh, I am so glad you brought that up because we absolutely have to clarify this. No, we cannot do that.
Why not?
Broad-spectrum antibiotics act like a nuclear bomb in the body. If you nuke this system, you aren't just killing the bad guys. You wipe out the beneficial bacteria too, like the natural lactobacillus that actually keeps the immune system healthy in the first place.
Right, you're destroying that helpful diversity we just talked about with the long-term survivors.
Exactly. Ruining that delicate immune homeostasis breeds dangerous antibiotic resistance. And ironically, it can actually worsen systemic inflammation, making the cancer's environment much more aggressive over time.
Oh, wow. So it backfires entirely.
Totally. The entire future of this field relies on precision. We have to use targeted antifungals and these engineered probiotic living drugs as a scalpel, not a sledgehammer.
A scalpel, not a sledgehammer. That makes perfect sense. Time for our final recap. High microbial diversity is actually linked to longer survival. And treatments now focus on precise antifungals, engineered Trojan horse living drugs, and fecal transplants to reboot the tumor's environment without blindly nuking the whole system.
That covers it.
It's just wild to sit here and realize that the future of treating one of the most impenetrable, deadly solid tumors on Earth might literally involve intentionally swallowing a genetically hacked version of E. coli. It flips everything you thought you knew about infections and cancer therapeutics completely upside down.
It really is a paradigm shift in modern medicine. We're looking at cancer through a completely new lens.
So let's pull all of these massive concepts together as we wrap up this deep dive. We started by looking at a supposedly sterile VIP room, only to find out the doors have been propped open the whole time.
By inflammation.
Exactly, and it is absolutely mind-bending to realize that a seemingly random, completely normal yeast living on your scalp right now, or a tiny pocket of bacteria tucked away in your gums, can traverse your bloodstream, slide past a malfunctioning bile duct, and literally rewrite your immune system's source code to build an impenetrable fortress for cancer.
And when you trace the trajectory of all this research, it leaves us with a truly profound, almost unsettling realization.
What's that?
Well, if our skin, our oral cavity, and our gut microbiomes are constantly migrating, constantly moving through those secret tunnels and seeding our deep internal organs, how much of our long-term systemic cancer risk is quietly, invisibly being dictated by our mundane, daily environmental choices?
Oh, wow. That is a lingering thought right there. And as you're listening to this, I really want you to reflect on your own microbial footprint today. Think about the specific brand of toothpaste you use. Think about the antimicrobial skin creams you apply every morning, or the exact composition of your daily diet. Are your choices accidentally feeding the microscopic masterminds that pull the fire alarm? Or are you actively cultivating a diverse, protective microbial armor that keeps those invaders in check? It's definitely something to ponder as you go about your day. Thank you so much for joining us on this incredibly dense, eye-opening deep dive.